Good afternoon, all of you. On behalf of SSR College of Pharmacy, I take privilege to introduce our next speaker of the day, Dr. Satish Panchal, sir. Sir is the current functional head of toxicology operations within Translational Development Department at Sun Pharma Advanced Research Co. Limited, a Sun pharmaceutical company in Vadodara. His group delivers support to GLP, non-GLP, and investigative toxicology and pathology for small molecules, peptide, biosimilar, and vaccines. Sir has pharma industry experience that spans more than 21 years with pharmaceutical companies and CRO. He has a strong scientific interest in testicular and female rate reproductive system toxicology and pathology. He has been supporting development of a novel approach to automatically change testis for spermatogenesis, estrous cycle, and ovarian follicle count in rat. He is an active member of Society of Toxicology Pathologists India, also acted as chair IFSTP webinar committee and participated in conducting and delivering talks at workshop, webinar, seminars, and conferences for STP Fund. Currently, he is a treasurer of STP Fund. He holds bachelor degree in veterinary medicines and master's degree in veterinary pathology from Anand Agriculture University, Anand, Gujarat. He is a diplomat of Indian Board of Toxicology Pathologists and UK registered toxicologist. We are so glad to have such an eminent personality. Sir will share his valuable knowledge on today's topic, peer-reviewed histopathology. Sir, you can proceed with the session.
Thank you, Bhavini, for the introduction, and thanks to Dr. Sanjeev, Dr. Chirag, and the organizing committee of the FDP program for giving me an opportunity to share my experience with you. And as my first slide shows that I'm going to talk about the GLP requirements for peer review of histopathology for the studies which are conducted in the non-clinical toxicology settings. So I wanted to be current with the presentation. Today is a World Pharmacist Day, that is 25th September 2020, and I would like to congratulate all the pharmacists for their sincere work and especially the work that is being carried out in the trying times of pandemic due to COVID-19. So this is my confidentiality disclosure statement. So we have to make this statement as a routine practice, because I am an employee of Sun Pharma, and I have to disclose that the ideas, opinions, and comments which are described in this presentation are my own and do not represent to those of my current work affiliation company, that is Sun Pharma Advanced Research Company Limited. And I approve this statement today and also release it today as I am presenting today on 25th September 2020. Next one, please. So as the topic of my talk is the peer review of histopathology, so I need to set a background that what is peer review pathology, why it is needed, why the OECD principles or OECD GLP has made a guidance document. So whenever any toxicology studies are conducted, so during the conduct of toxicology study, we monitor several parameters during the conduct of study and also at the end of study. And one of the parameter is a histopathological assessment, which is a key endpoint for the toxicology study. So what is histopathological assessment? It is at the end of toxicology study, we collect various tissues, organs from lab animal after sacrificing them, and the collected tissues are preserved, processed, and histology slides are prepared, examined by pathologist, and result or data is generated. So that is what is called as histopathological assessment. And the data generated would reflect either the drug which is given to animal for a stated period of study duration, whether it has caused any toxicity to that organ. And the results contribute substantially to the outcome and conclusion of the study, as any drug which is affecting the organ would also a signal for clinicians when they conduct clinical trials of any new drug. So clinicians need to know what are the toxicities observed or reported in various toxicology studies before clinical trials are initiated. And the histopathological assessment, which is done in a toxicology study, is done based on the opinion of a single pathologist. As observation of tissues and various organs under the microscope is really challenging and a bit tricky kind of exercise, it is always recommended that the pathologist who has conducted original examination has to get an opinion from a second pathologist. And second pathologist is called as a peer review pathologist. Because why the peer review is needed? Because before it is submitted, a company or organization need to make sure that histopathological findings examined by single pathologists and reviewed by peer review pathologists are meeting the regulatory requirements. And once the peer review process is done, it also leads to changes in the interpretation and the reported results, and at the end, it can potentially change the outcome or the conclusion of the study. Hence, the process of peer review is very, very important. Next one, please. So who should be familiar with the peer review process?
No presentation is being visible, sir. Excuse me, sir. No presentation is visible for us, sir. It is blank showing.
It is visible. Kindly check your internet connection.
It's visible, sir.
So who should be familiar with the peer review process? So until now, you must be familiar with the terminology like pathologist, peer review pathologist, and test facility management, study director, quality assurance personnel, as the program is specifically focusing on the GLP principles, and this program is running since 22nd. So you all know what is test facility management, study director, and QA assurance personnel in GLP. So when peer review processes are to be conducted, pathologists and peer review pathologists should be familiar with the GLP principles, test facility management, study director, quality assurance personnel, and these are the familiarity is needed in order to meet the GLP expectations and requirement. And many times it has been observed that sponsor here is the organization which awards study to any contract research organization. So the chemical belongs to sponsor and the study is conducted by a contract research organization. So the owner of a drug is called a sponsor. So in many situations, sponsor wants that peer review to be done by specific peer review pathologists. And why specific peer review pathologists? Because they are the expert in the field. Like there are several peer review pathologists existing in the world, but everybody has their own expertise depending on the system. So some peer review pathologists, either they are expert of respiratory system, some are expert for the male reproductive system, some are expert for lymphoid system, immune system expert. So sponsor knows that if there are any changes related to particular organ system, then peer review pathologists should be accordingly identified and appointed to conduct the peer review examination. And by doing so, it will allow the consistency of finding and terminology or diagnostic terminology used in the study, and also the interpretation through the study would be consistent. And if the expert may not be working in a GLP facility or they may not be an employee of the GLP facility, but it may not be always possible that peer review should be a GLP compliant. So the next slides would also help us explain that in case the peer review pathologist is not working in GLP compliant facility, even then how to make the peer review process GLP compliant. Next one, please. So before performing the peer review process, peer review processes are to be clearly described in the study plan or study protocol. And if for any reason it is not described in the study plan, then it can also be added by taking the study plan amendment or study protocol amendment. Normally, OECD principle uses study plan as a terminology, but very frequently protocol is also being used by many investigators. So the peer review should include the information on how peer review will be conducted, that is how peer review will be planned, managed, documented and reported, and performed. So the peer review can be done contemporaneously or retrospectively. And if you have SOP which describes how the peer review will be conducted, then in the study plan you need to just give a reference that peer review will be done according to this SOP. And SOP should clearly describe that reconstruction of the peer review process, how it will be done, or how the tissues were selected for peer review. And majority of the times, when peer review is done also to confirm that the tissues which are showing changes are in concurrence with both the pathologists, that is study pathologists as well as peer review pathologists. Hence, in the study plan or in SOP, you need to define how the tissues will be selected for peer review. So I'm also going to talk about the SOPs for peer review in the coming slides. Next one, please. Yeah, this is the flow diagram, how the peer review process goes on. Like once the slides are ready for the examination, slides comes to pathologists, pathologists examine the slide, they record their result or their result output in a format, and depending on the integrity or requirement from sponsor, peer review can be done either formal or in informal way. So informal way is like study pathologists seek view from other pathologists before concluding the diagnosis and finalizes pathology report. So for an example, in informal pathology peer review, if I'm a study pathologist and I have a doubt or any lesion or change, then I will seek the opinion from my senior pathologist or colleague pathologist. And if we are in agreement, then it's an informal way. So we need not go to the formal way of a pathology peer review. So informal way, I can just take an opinion of another pathologist and finalize my diagnosis. But in case of formal way, there are two ways, like either you do internally or externally. For internal peer review, you have a pathologist, senior pathologist, experienced or expert pathologist within the GLP facility employed by the organization. And he or she conducts peer review. And in similar way, when it is external, so company hires external peer review pathologist, and then depending on the timing, whether it is to be done prospective or retrospective, it is further processed. So in case of prospective peer review pathology, expert pathology, they conduct this slide examination before the pathology, that is study pathology, finalize the pathology report. And if needed, if there are any changes needed, then follow the study plan and take the amendment and perform the peer review. And in case of retrospective peer review, the pathology peer review is done after study pathologist has signed this anatomy pathology report, but the study director has not yet signed the report. So in that case, retrospective peer review pathology to be done. And where needed, the amendment can be done from the study protocol and perform the peer review and amend the pathology report of study pathology. And in case if the study director has also signed the study report, then the peer review to be initiated by sponsor and test facility by taking an amendment of the protocol, perform the peer review, and amend the pathology report and also the study report. In case when regulatory, where the report has been submitted and the regulatory agency asks to peer review the findings, then follow the regulatory instruction, perform the peer review, and then prepare a separate report and submit it to agency. So this is in a single diagram how the entire peer review process goes. It very clearly describes, and there are number of publications available on pathology peer review. I can even share later on if somebody is specifically interested in any part of the peer review. And can you go to the next slide, please? Yeah. So GLP requirement continues that if the peer review pathologist is located remotely from the study site, that is, for an example, the study is carried out in Vapi and the peer review pathologist is located in America, then how the peer review process should go. So in that case, when the peer review pathologist is not available on site, then it is not necessary that peer review pathologist to be appointed as a principal investigator, because peer review pathologists generally they do not generate data. They just review the data generated by study pathologists. So peer review pathologists can be considered as a contributing scientist and not a principal investigator. But the major responsibility lies with the study director to ensure that peer review process is conducted in accordance with the principles of GLP. OECD has a clear-cut guidance document that is of 11 pages, was published in 2014, and worldwide it is being followed by all pathologists or organizations conducting the peer review. And how the peer review process was conducted, that also to be documented and to be retained within the toxicology study files. And this activity will also include on the identity of the tissue, that what are the tissues that were reviewed by the peer review pathologist, when it was reviewed, and by whom. So all the documentation has to be done, and study director has to ensure that everything is done and documented appropriately and as soon as possible. During the peer review process, when peer review pathologists generate notes, so those notes are normally not retained in the study file. It is not to be retained in the study file, but a peer review pathologist need to generate a statement that he or she has examined. Next one, please. And each correspondence happening between the sponsor, test facility, and peer review pathologist should be retained in the study plan. So correspondence could be either minutes of teleconferences, email communications, or any other hard document exchange that has happened among them. And all the slides, corresponding blocks, which were used for preparation of the slides, to be archived. And the archival duration should be the same as to that of other raw data that were generated and archived from the study. So purpose of archival, as all of you must be knowing, that the purpose of archival under GLP condition is for the purpose of reconstruction. So reconstruction here is like experiment we have done, everything has been recorded, reported, and archived. So in case in future, if any query arises or in case of any further investigation, all the documents, including slides, blocks, and tissues, should be available for reconstructing the data that have been generated during the study. And there are many instances that study pathologists and peer review pathologists, they do not concur or they do not agree to the conclusion drawn by the original pathologist. So in such cases, a clear, transparent, and unbiased process should be implemented to resolve the differences, as histopathology examination is very, very subjective, and it is a very challenging and tricky situation when it comes to the level of peer review of histopathology slides. And every time it may not happen that study pathologists and peer review pathologists would agree to each other's observations or changes or lesion identified by them. So in case if there are any differences arises during the study peer review process, then it has to be very, very clear and transparently documented. And these procedures are also to be documented in form of SOPs. Next one, please. And in cases where peer review pathologist findings are significantly different from the original pathologist finding, then it also needs to be addressed or documented that how differences of interpretations were handled and changes made to the study pathologist's original interpretation should also be described in the final report. So as I said in the previous slide that it is not always that pathologists would agree with each other's observation, but they need to arrive to a consensus and agree to the finding, and the agreement has to be reported in the final report. If still the differences are there, study pathologists and peer review pathologists have not achieved an agreement, then in that case, an independent expert or panel of experts should be appointed to resolve the issue, and the conclusion of the panel should be clearly documented in the final report. I have one picture to describe this, that how such differences are to be addressed. It will be on the slide number 20. I will explain you there also.
Presentation is not visible. Can you just check? Slides are not visible.
It is visible. It is visible,
No,
sir. Kindly check your internet.
It's okay.
It is visible to us, sir. To all, sir. Sir, please continue.
Yeah. So, but in majority of the cases, if there are no significant differences of the opinion occurs during the process, then it is not necessary to report in detail the outcome of peer review and the pathology report or final report. Just a simple statement from a peer review pathologist that would suffice, and that can be a part of the study file, not the part of the study report. Next one, please. And there are no requirements for peer review pathologists to sign the final report, but there is an expectation from OECD that peer review pathologists should sign a statement, and the statement should be retained within the study plan, and that will help us to prove the identity and affiliation of peer review pathologists, and that also has to be a part of final report. So final report, there is no need of a statement from peer review pathologists, but the name, that is identity and affiliation of peer review pathologists, should be reported. Next one, please. So until now, what I was describing was the GLP requirement. Now, how to comply with the GLP requirement. So GLP compliance of peer review. As I mentioned, that peer review process can lead to changes in interpretation of histopathological finding, and in turn, it may influence the outcome and conclusion of the study, because It is directly indicating whether the drug is causing damage to the organ or not. So it has to be very, very clearly described in the report. And peer review process can lead to change in the conclusion after the peer review is completed. It may not always happen, but it may happen. Hence, it is expected as per OECD principle that peer review should be conducted in compliance with the GLP. As peer review has scientific value and appropriate specialist to be appointed with his expertise for conducting the peer review. And many a times it happens that the peer review pathologists are not working with the GLP facility. So in that case, if peer review is done in non-GLP facility, then we need to justify and record within the study plan and final report. But to meet the expectations of OECD and to conduct the peer review in GLP environment, still we should make an attempt to conduct peer review in GLP way, and how it can be done when the peer review pathologist is not working in the GLP facility. So in such cases, you do not need to transfer histopathology slides from one side to another, but allow the peer review pathologists to perform their work under established GLP quality system by training them with the relevant procedures of the test facility. So OECD principle clearly says that it should be done in GLP. So that is the expectation of OECD, that peer review should be done in GLP. But when pathologist is working in a facility which is not GLP, and in those cases we need to transfer, we may not transfer slides to them, but in those cases we can just train them in such a way that it leads to compliance. Next one. Here, study director to make a statement to which extent the GLP compliance was followed while conducting the peer review, and ultimately the responsibility lies with the study director for overall compliance of the study. And in case if non-GLP site was used for peer review, study director needs to clearly describe in the study report, and during the process of peer review, study director has to ensure or has to gather enough information about the peer review pathology, like what is the experience of peer review pathologists, what is the expertise of the reviewing pathologist, a review of the test facility SOP or documented agreement that peer review pathologists will use the test facility SOPs and procedure. So you need to generate a documented agreement that peer review pathologists will use the procedure which is established by the test facility. And study director also need to ensure the chain of custody of the samples and associated paperwork, security of samples and documents while at the peer review pathologist facility. And in case any computer system used for data capturing, like currently Proventis, IBM, PDAS, there are several other software solutions available for capturing the histopathological finding using the computer application. So if a peer review pathologist is going to use that, study director should ensure that it is properly validated before use, and also adequate quality assurance activities, which may include an audit of premises and equipment used by peer review pathologist. So if peer review pathologist is using his own instruments, then there has to be some quality assurance activities performed there by quality assurance department. And if the peer review is conducted in a non-GLP facility, then it should also be described in the study director's statement. So in cases if it is not at all feasible for peer review pathologists to come to the test site or test facility for conducting peer review. In any case, peer review pathologist has to perform the peer review in non-GLP condition. Then study director, when he or she finalize the report, in the study director statement, they have to very, very clearly mention that this phase of the study was non-GLP. So regulators, when they review, they would immediately understand, and if needed, they can recommend further review under GLP condition. Next one, please. So summary of expectations. So overall, what is the expectation from OECD? As the peer review of histopathology is an important part of the process, which ultimately affects the overall outcome of the study. Hence, peer review has to be done. And it is essential that peer review procedures are planned, conducted, documented, and reported in such a way that integrity of the regulatory study is not compromised and activities can be fully reconstructed and verified. That is what I was explaining in one of the previous slides. That reconstruction of the results or data is very, very important aspect of GLP. So whatever the data that have been generated, documents have been generated, everything has to be archived. Even the tissues collected, samples collected during the studies, everything has to be archived. So in case needed for any data reconstruction, we can just collect the data from archive or samples from the archive and then reprocess and examine. Histopathology peer review activities should also be described within the study plan or subsequent amendment. That's also one of the expectations of OECD. And it should also describe the tissues and documents examined by the peer review pathologists, and the reporting of peer review should be sufficiently detailed to allow reconstruction of the process and the opinions expressed. There should also be a documented procedure, or during the peer review, it should be documented if there were any differences between study pathologists and peer review pathologists, so how the opinions were resolved should also be documented. And if still differences are not resolved, the interpretation of that result in a significant change of the study pathologist's original interpretation, then that also needs to be discussed in the final report. And it is very, very important to report the identity and affiliation of peer review pathologists in the final report. So these are the expectations of OECD in brief, that how the peer review has to be done and how it is to be reported in every situation, whether both the pathologists are in agreement or not in agreement. Next one. Now, a few slides will be there just to recapitulate why peer review is required. So peer review is required just to ensure the accuracy of data, to ensure that data meets regulatory agencies' expectations, to confirm target organs. What does it mean by to confirm target organs? So original pathologist, when he or she has examined the slides and found that kidney shows some lesion which could be due to the drug or test item which have been administered to animals. So in that case, kidney becomes a target organ. So whatever changes or lesion reported by original pathologist to be validated or reviewed by peer review pathologist. So by this way, it will be confirmed whether the original pathologist finding are matching with the second pathologist finding. So that way you can confirm the target organs. And also to confirm NOEL. NOEL is no observed adverse effect level. So based on the findings, study director would declare NOEL based on the overall data and effects on various parameters that have been monitored during the study. So if peer review is done, then that can also help in confirming the NOEL. And this will also ensure that diagnosis or changes which have been reported in the study which has got a consistency. There is no change from pathologist to pathologist. And sometimes it is also done as a part of continuous education. So junior pathologists, when they are examining the slides for the first time or in their early career, then peer review would be a part of a continuing education for them. And that will also ensure the quality and the accuracy of the interpretation. And it's also help to maintain the best practices. And peer review is also done in case any specific issues are to be addressed. Next one, please. So how the pathology peer review is to be done? So it is to be performed by a second pathologist who is expert and well experienced. It is routinely performed for the non-clinical toxicology studies conducted globally. So I would also recommend that whomsoever will be going into a toxicology CRO or CRO where preclinical studies would be done, and being a study director should insist on peer reviewing of histopathology slides, whether it is a single dose study or subacute study, subchronic study, carcinogenicity studies, whatever it is. Peer review will also include the representative tissues from study pathologist evaluation. So peer review is not a reread of a study. So it is not the entire study which is being read by study pathologist will be read again by a pathologist. So it is not so. And it does not generate a second data set. That means that whatever the data that have been generated by original pathologist would remain as it is. But after peer review, if there are any changes to be done based on the opinion of the expert pathologist, so that data will be updated. So it is not that data are generated again. And peer review is never done in a blinded way. What the blinded way means, let's say for a 30-day study, we have 3,000 slides to be examined by pathology. So it is not that each slide to be blindly coded. They will be given a code so that pathologists will not know what this slide is, from which animal, which group. So it is not to be done in that way. So it is to be done in unblinded way. And many times it happens that especially for a junior pathologist, and when peer review is to be done, so they undergo kind of anxiety or agony, think that it's going to be a part of my performance review. So it is not so. It is not a performance review of a study pathologist. Next one, please. Yeah. So this is what I was saying in one of the previous slides, that there has to be SOPs, that is standard operating procedure, how the peer review process would go. Whatever I have explained until now is OECD requirement, and how to comply with the OECD requirement and what are the expectations of OECD guidelines. But it is up to the institution to design an SOP and implement. So how the SOP has to be designed. So globally, even there are publications available for the best practices for conducting peer review. So normally when we do peer review in our organization, we take animals, representative animals from control and high dose group. That is for subacute, that is up to 30-day toxicology study, and up to 90-day toxicology study. We take 30% of animals from control group, and we examine all organs and tissues from those 30% animals. And similarly for high dose group. If it is a carcinogenicity study, we take 15% of the animals from the study. So if any carci study would have, if 50 male and 50 female, that is 100. So we take 15 animals and examine all 15 animals, all organ tissues. If it is a dog study, then 100% of animals will be examined from the control group. And in case of non-human primate study also, 100% of the animals are examined. Why in dog and non-human primate animals we examine 100% of animals? Because here the number of animals per study are very less. So if I say we do a 30-day and 90-day tox study in dog and monkey, would have 3 male or maximum 4 male, 4 female per group. Whereas in case of rodent, up to 90-day toxicity study would have 40 animals. And there it may not be feasible to examine all animals. So since the population or number of animals employed in the dog and primate study are less, we examine all animals, all tissues. Next one. Yeah, so in case we observe any mortality during the conduct of study, so to identify or verify the cause of death, selected tissues from all animals that have died during the studies are examined. This is also one of the part of our SOP. Or institution can decide based on their policy how many to be. So it depends from organization to organization. But peer review processes are to be done to meet OECD requirement. In case there are any target organ, then examine all target organ from all groups. Here all groups means control, low dose, mid dose, high dose, and if you have a reference group, then from all groups you have to examine target organs. And that will help you accurately confirming the NOEL. And in case you are doing a long-term studies like 6-month and carcinogenicity studies, then in those studies, all tumors or neoplasms from all those groups, low, mid, high control, negative control, and proliferative changes. So proliferative changes are mainly hyperplasia, hypertrophy kind of thing, which are non-tumor changes from all those groups has to be examined. Next one. Yeah, so this is very interesting part. What are the reasons of disagreement in pathology review? As I was explaining in a few of the previous slides, that study pathologists and the peer review pathologists, in many incidences, their opinions do not match. They do not agree. So what are the reasons why they do not agree? What are the reasons of difference of opinions? So every pathologist, they use different criteria for classification of tumor. I would use different criteria for classifying the tumor, either it is adenoma or carcinoma. And the threshold which is used for diagnosing the lesion are also different from pathologist to pathologist. So here the severity, I would say that liver is showing the infiltration of MNC, which is of mild severity. But the same slide, another pathologist would say it is of moderate severity. So these are the reasons why there is a disagreement in the pathology review occurs. Sometimes when lesion is not, the pathologist is not familiar with the lesion or lesion is unknown, and in those cases also disagreements arises, or the different terminology is used for same lesion. So that also happens. And there are also differences in diagnosis. So these are some of the reasons I have listed why the disagreement takes place. But how to address? This was a global challenge. So how to address these differences? So there are several societies existing, like Society of Toxicology Pathology India, Society of Toxicopathology US, Japan, British, Europe. So all three societies have come together, and can we go to the next slide that I can explain you there? Yeah. So here, this is what I was telling, that there is a slide number 20, and in cases where study pathologists and reviewing pathologists do not agree, then this is the workflow, that study pathologists would examine in unblinded way initially, then it will go to the reviewing pathologist, that is peer review pathologist, in unblinded way, but still the agreement is not achieved. Then in that case, pathology working group, that is PWG, is to be appointed. So it is a panel of experts who would examine the slides in a blinded way, and they give their consensus of diagnosis. So after that is done, then the final data set is generated. So there may be instances that peer review pathologists and study pathologists do not agree with each other, and in those cases, panel of pathologists has to be appointed. But this situation comes very rare. Yeah, so this is what I was explaining, that it's a global challenge, and every pathologist were explaining the reasons or findings on their own way, and those were the reasons why the differences were arising. So the number of societies have come together and they have designed a criteria. It is called International Harmonization of Nomenclature and Diagnostic Criteria in hand, which is very popular among the pathologists. So what I have listed here, there are all in-hand documents available for almost all organ systems for rat and mouse. Similarly, rabbit, dog, monkey, and other species are also being drafted and will be coming soon. So pathologists, any pathologist, when they read the slide, they will have to follow the criteria described in this guidance document. So the lesions are described uniformly. Next one. Yeah. So as pathologists, when they present, their presentation normally doesn't get over without showing any images. So I am going to show you why the peer review of pathology slides are needed. On the right hand side, you would see that this is a kidney. It's a normal kidney from rat, and left image shows the chronic progressive nephropathy. So what happens if any pathologist is seeing the slide, and if he or she is finding chronic progressive nephropathy, normally we have experienced that they do not describe it as a chronic progressive nephropathy. So they would describe that there is a tubular atrophy, there is an interstitial fibrosis, there is a hyperplasia, there is a dilated Bowman's capsule. But when it is subjected to peer review pathologists are expert pathologists. They know how to uniformly describe each lesion. So it is to be described in a simple and very, very conveying manner. So for this kind of change, it is called chronic progressive nephropathy. And whatever I have described below the left image, it overall describes what are the changes in that kidney image. And that is the reason peer review of pathology slides are to be done. Next one. So this is the normal testis from rat. Right-hand side is a normal image. Left-hand side is an angiectasis. That's also testis from rat. So angiectasis is a focal dilation of blood vessels. So what has happened here is the seminiferous tubule is completely occupied by the blood components. But when the study pathology is observed, sometimes they have a difficulty in diagnosing lesion, either to call it angiectasis, either to call it as a hemorrhage. So there are different terminologies, or edema, whatever it is. So when pathologists have a second opinion, then they can definitely confirm that it is angiectasis. And this is how these are all changes needs to be peer reviewed. Next one. This ethylene glycol with EGME, ethylene glycol monomethyl ether induced testicular toxicity in rat. The center one is a seminiferous tubule from testis, and it shows that the testis is such an organ that it undergoes number of physiological changes. And when pathologists review, even for pathologists also, it becomes difficult to identify any changes that occurs in the testis, especially the spermatogenesis aspect of it. There are total 14 spermatogenesis testis, 14 stages of spermatogenesis in rat, and there are certain drugs which affects various stages of spermatogenesis. So here in this image, it is a stage 4 to 5, where you can see the elongated spermatile, especially the cytoplasm of elongated spermatid is forming a vacuolation. So those vacuolations can be seen within the lumen of the seminiferous tubule. But these are very subtle changes which normally not easily identified by the pathologists unless and until pathologists are very highly trained or experienced. So to identify such subtle changes, peer review processes are must. So I would strongly recommend that anybody of you, if you are going to join CRO or in future, or you are already associated with them in some or other way, then make sure that peer review of histopathology slides are always done. It's very, very important. Next one. Yeah, this is another change which is induced by EGME. So here you can see that tubules are being, most of the round spermatids are depleted. I do not have a cursor here, as I could have shown you, but definitely you can see that some of within the seminiferous tubule there are empty spaces. So those empty spaces are nothing but the degenerated round spermatids. But these are also very subtle changes. So unless and until the pathologist is experienced and well trained, these changes normally goes unnoticed. And when the drug is moving from preclinical to clinical stage, and when something happens during the clinical trial, then regulators, they recommend that you go back and see your animal slides, what has happened there. So, and during that process, organizations lose a lot of time, money, and resources. So it is always good that peer review of pathology is done prospectively. Next one. This is same EGME induced, and here in the within the seminiferous tubule, you would see that there are elongated spermatids, but not in as many quantity or volume as it should be, because EGME is one of the toxicant. But pathologist has to see it very carefully and also has to compare with the control. Then only these subtle changes would be identified. Next one. The same one, like Sertoli cell vacuolation. So here you would be able to see in the center, there is one seminiferous tubule, and at 11 o'clock position, you would be able to see some vacuolations. So these are the Sertoli cells vacuolations, and also there are no elongated spermatids within the lumen. So this is all toxicity caused by the ethylene glycol monomethyl ether. Next one. It's a normal liver from the rat on right-hand side. Left-hand side is the liver, the fatty liver, because of chronic alcohol consumption from rat. So it's an experimental model. So what typically happens when alcohol is being consumed for a long time? So hepatocyte, that is cell from liver, are being replaced by the fat. And as the lesion progresses and condition deteriorates, the liver cells are being replaced by the fibrous tissue, and that condition is called as a cirrhosis of the liver. And once the cirrhosis occurs, then it is irreversible, and commonly also it is known as a liver cancer. So you can see the difference, but unless and until it is peer reviewed and or the second opinion is sought, it is difficult to interpret in the report. So it is always good that second opinion from the peer review pathologist is obtained. Next one. In the current scenario, globally, what is happening is now many companies, they are digitalizing their slides so that peer review can be done from anywhere. I am sitting in Vadodara. We have a scanner. I scan the slide, generate the digital image, and then same image can be shared to anybody sitting anywhere in the world. So in the real time, I can get the second opinion. And that can also be done under GLP condition. And there are also number of microscopes available. There you do not need scanner. Just connect your microscope to internet and it will be like I am seeing in the microscope and you would be seeing on your screen. If I move the slide, you would see that slide is being moved. So it's a real-time slide observation can also be done when such microscopes are used. So that can also help you conduct the peer review in very faster way, like especially in cases when you have peer review pathologists from outside your country. And this has become a now very frequent and regularly used. Next one, please. So during the COVID time, US FDA, United States Food and Drug Development Agency, to meet the emergency, public health emergency, they have published this guidance document. They have enforced this policy to remotely conduct digital pathology devices, use the digital pathology devices during the coronavirus disease. So this is being implemented and will be effective until the corona situation resolves and to meet the emergency, public emergency. So I would like to conclude my presentation with 2 more slides. This is the human lung, which is normal. You can see the alveoli here. There are blood vessels, bronchioles, capillaries. It looks very normal, healthy. Alveoli are dilated, open. So this is a normal lung. Now I'm going to show you a lung from corona patient. So this is the histopathology image of a lung from a patient who suffered from COVID-19. So if you compare this slide with the previous one, you can see that slide A here has got number of inflammatory cells and alveolar damage, even hyperplasia of alveoli. There is a hemorrhage. On the B part of it, there is a thrombi within the artery. C part also shows the thrombi and also the D. So this is what happens because of corona infection, that it completely damages blood, lung, causes the thrombi, that is a coagulation of blood within the vessels, and then patient die because of respiratory insufficiency. Next one. These are the references I referred for preparing my presentation, and the best reference among all is the OECD principles, because to match the theme of the program, it's the OECD principles of GLP for peer review pathology. So that's all from my side. If you have any question, maybe I am happy to take the question now.
Yes, sir. Yes, sir. Thank you, sir. It was our privilege to learn from you the topics related to peer review of histopathology, role of peer review pathologist, GLP compliance, some SOPs, and the best part was you have shown the peer review example slides. So, sir, with your permission, I would like to take some questions from the participants.
Sure, I'll be happy to answer.
Okay. Okay. Dr. Suvendu Kumar Sahu, he has the question: can histopathology be a good career option for students?
Yes, it is a very, very good option. And there is a scarcity of pathologists globally, not only in India. When we talk about the toxicology pathology, I think within India, who would be practicing toxicology pathology would not be more than 100. So globally, there is a dearth of pathologists, and it's a very interesting, but if you are ready to take the challenge, it is the field you should go for it.
Okay, sir, what are the qualifications required for this?
So to become a pathologist, I mean, you need to study bachelor in veterinary science.
Okay.
And after bachelor, there is a specialization program, that is veterinary pathology. That's a 2 years program with thesis, research thesis to be completed. And then you still further study the PhD also in that area. So the veterinary minimum qualification of Bachelor of Veterinary is must, and then Masters.
Okay. Sir, Dr. Javed Ahmed Khan, he has the question that artificial intelligence and machine learning tools, can it be included in advanced histopathology as per GLP guidelines?
Yes, it's a very good question and very timely. And this is the time where whole world is working on artificial intelligence, machine learning, deep learning, and I'm happy to share that I'm also associated with several programs of this kind, and we have developed even automated staging solution. So when I was explaining during my presentation that there are 14 stages of rat spermatogenesis, so we have developed in-house developers software solutions which will automatically stage the seminiferous tubules in different stages, and that is being developed based on the artificial intelligence, machine learning, deep learning. So several other organizations are working. People are coming up with the different solutions. So it's a time for digital pathology.
Okay. Sir, Abhishek Arang has the question. What is drift diagnosis?
Sorry, I did not get.
Drift diagnosis. What is drift diagnosis?
Drift diagnosis, yeah. So drift diagnosis is something like if I am examining the slides and I am seeing that there are necrotic foci in the liver, but out of 10 animals, I see the necrotic foci maybe only in 2 animals. But when it is also seen, those slides are seen by peer review pathologists. Peer review pathologists would say it is not 2. There are 8 slides with necrotic foci. So then there is a drift. I say it is 2. Peer review pathologist says it is 8. So there is a drift in diagnosis. So only the number changes there. And that is the reason why peer review is very, very important.
Very
So
essential.
unless and until you so when you submit your report to regulatory agency and report says that slides have been peer reviewed, that's enough. Most probably you will not get questions from them.
Yes. And sir, one question is from Preeti Zidi, but what I understood from her question is she is asking that the control, for the control which standards we are using, from where to procure that?
The control, if you are using for toxicology testing, then control in our condition, it is a drug which is already approved and available in the market. So either you have to get it from domestic market, that within India. If it is not available, then you need to get it from the appropriate country where it is marketed and available. So then you have to undergo several legal procedure to buy.
Okay. Sanjeev, sir.
Yeah, there is one question I have, that if the drug is already approved by some FDAs, US FDA or MHRA has approved, then how much this particular studies being required, this peer review and everything? Is it a first time study required, or even at a formulation level also these things are required to be done? API and formulation, if we are having 2 things, then whether both things require this is a mandatory?
So peer review is required whenever you are doing toxicology study.
Okay.
So in case if the drug is new and you are doing toxicology study, then it is to be done. If drug is already available in market, but some companies, they repurpose the drug or reposition the drug. So for an example, like earlier medicines were available to take 3 times a day. But now formulation have been improved. So instead of 3 times a day, now we are taking only 3 times a day, once a day.
Okay.
So in those cases, when toxicology studies are required by the regulatory agency, it is to be done. So peer review is to be done whenever you are doing toxicity study.
Okay. Thank you very much, sir. It was a wonderful session. I think till date we have understood the histopathology. After your session, it's the entire big umbrella of we can say science is there in even doing. Sometime many pharmacology students are doing it, and many time publishing it. You may have realized, but the depth it is required is somewhere missing in our studies. And from your presentation, it was very clear and very understandable that you should have 3-4 opinions for one same observation. That is a major lesson which we have taken from your session. That's wonderful, sir. It was really we can say made clarity on many such aspects which we are doing, but we are not knowing how to perform them. So thank you once again from entire our team of organizers that you have taken this pain and given that 1 day time, almost a 2 hours time with us. Thank you one and all.
So thank you.
No, thank you, sir. We'll be in touch, sir. We'll wish you sometime you can visit our campus when the things get normal.
Sure. Looking forward to. Sure.
Yeah.
All the best.
Thank you.
Bye. Bye.
Thank you all the participants. We will see you tomorrow morning by the link provided in your WhatsApp group as well as email. Thank you. Have a good day.